Okay. Hi, everyone. I'm Sophie. I would, in fact, like to share with you some computational insights in protein adaptation across environmental gradients and also how I got to them. Um, so we define the environments usually by a set of diverse parameters, for example, the easy ones, temperature, pH, salinity if you work in water. Um, these are rarely ever constant in nature. They change by the hour, they change by the day, they change across hundreds of millions of years with climate change. So there is a constant pressure to adapt at the molecular level. The environment affects all levels of the cell metabolism. Some are transient, like the expression of heat shock proteins, while others get permanently recorded as mutations in the genome sequence. On the level of sequence, there exists a lot of models, tools, predictors, algorithms that have been developed and established to describe how this evolution of a sequence works and to explain a bit. But especially for protein sequences, making proteins the workhorses of the cell, there's a lot of additional constraints on the evolution that apply. These properties are usually encoded but not directly obvious from the sequence alone. There's a lot of predictors now that help us with this as well. A classic example of this is protein folding, where from a sequence that holds all the evolutionary information, we can go to a 3D shape, a static image of this fold. To me, protein structures are more the result of local interactions defined by the biophysical properties of the sequence. Those interactions are usually transient. So a protein isn't really a stable thing. It tends to move around sometimes. Hence, I think a probabilistic view of a protein on a per residue resolution is very interesting. These biophysical behaviors tend to also be more conserved than the sequence itself, which is very helpful if you're looking at involution. However, the sequence does carry all the evolutionary history with it. It contains redundancies to maintain a functionally important fold. Some sequences can fold into multiple different structures, which are difficult to capture by structure prediction or experiments. So my first goal in looking into protein adaptation was to capture as much as possible both the sequence and structure information in a format that works with all the tools that I have learned about. So in the past years, me and a colleague, Charlotte Kraus, we developed SimSAPiper, which is a pipeline to create compact, high-fidelity multiple sequence alignments on a scale of protein families, so a few thousand proteins, using well-established tools like tCoffee and MUFT and some maybe clever pre- and post-processing. We released version 2 of SimSAPiper a few weeks ago if you want to check it out. The best part about SimSAPiper is that it takes a lot of the work out of creating structural alignments because it collects structural information on its own or predicts them if you have the resources for this, and then creates structure-informed multiple sequence alignments. This is an example that I'm going to demonstrate a lot of the downstream work on. It's a aldolase family protein and a Timbal fold. It doesn't have too bad sequence similarity, but you can clearly see that while the sequences varies, the secondary structure stays firmly conserved. And this very compact alignment lends itself to a lot of the downstream tools because every time there's a gap, there tends to be problems. So these are our priorities for SimSAPiper as well. With this alignment, we can now employ a host of computational tools. First of all, what we are likely to do is to map this amino acid alignment onto the nucleotide sequences. And then, for example, we can look at the evolutionary pressure along this sequence. Here I am showing the synonymous over non-synonymous substitution rates between the nucleotide sequences. Essentially, this method considers synonymous mutations as the background that doesn't have a functional effect, and then the non-synonymous ones would have the potential to have a functional effect. And it expresses sort of if areas of the gene are changing more than it was normal. And that is called evolutionary pressure. So in this case, there's only a relatively weak signal for positive selection, but it's interesting nonetheless. Another thing that I can do with such alignments is create phylogenetic trees. These already reveal a host of interesting facts. For example, that the evolution of a protein is not the same or doesn't have to be the same as the evolution of an organism, but also they don't exist in a vacuum. You can see in this specific tree, based on the color of the dots, they represent the temperature at the sampling site. That even though some of my organisms come from very different temperatures, from very different locations as well, they cluster relatively closely in this example. What we can also do is predict a host of biophysical properties using the Bio2Byte tools, which is a tool suite that is developed in the lab of one of my supervisors, Professor Franken. And here I have a big green arrow pointing towards an interesting separation in the data that we can relate to a cluster of temperature similar, like a branch in the tree. So we can connect here for the first time the evolutionary history to like a biophysical expression of difference. And this area is consistently different if you consider only this small group of proteins. So here you can see a deviation from the colored sequences from the average, the background of this orthodox group. This one considers beta sheet propensity. We can see a similar thing in alpha helix propensities. So there's a shift where it's less likely to fold into a beta sheet and more likely to fold in an alpha helix at a certain location. These are just descriptive things that I can analyze from. Another thing that is very interesting to look at is the question of shared ancestry versus shared environment. Like, what could be the cause of this change? And in this example, it's very extreme. There's a strong correlation and anti-correlation pattern with the temperature. So according to this phylogenetic independent contrast, every single change in these sequences relates to the temperature. I was a bit worried because of the strength of the signal, so I also looked at the covariation of these properties, which also shows a strong pattern of like positive and negative covariance, which means if compared two residues in the pairwise comparison, if one of them has a stronger propensity, the other one also has a stronger propensity, that would be red, or lower, that would be the anti-version of this. And finally, I also looked a bit into the background of this specific protein, where at this position that we observed to have a possible change in the fold, it is close to the active site, which the active site is very conserved. The residues at this area don't change all that much. We can see a change in biophysical propensities, though, in relation to the temperature that I found these organisms in. The very interesting thing of this method is that it scales very well. You can draw it up to compare a whole proteome worth of data or multiple proteomes actually, because I'm looking at ortholog proteins. And you can scale it right back down and then look at the individual ortholog group or residues again. So that makes it really interesting to look into sort of great contexts. So while my work is mainly in the context of mesophyll or marine bacteria, it can be applied, and I have applied it to a wide range of data sets and organisms and properties, not just temperature, also salinity. As long as we can assume the orthology of the proteins, essentially, we can investigate the proteins with this method. And this analysis pipeline is capable to capture subtle changes that indicate protein adaptation in like a protein adaptation to environmental changes. Provides both per residue and per sequence analysis, and again, across a whole host of scales. So I can show you that structure-informed alignments are very powerful in looking at phylogenetic investigations, and that biophysical behavior is also very useful to get a better understanding of evolution. And with that, I would like to thank you for your attention. Thank my groups that are always here for discussions, context, and feedback. And I'm happy to take any questions.